So in section 15.5, we're going to look at step 4, and this first part here is the T cell response. And so we'll look at the main functions of the major T cell types and their subtypes, and the role of cytotoxic T cells in apoptosis. So a lot of this we've kind of gone over. We've seen several of these, and so we're just digging in deeper into step 4. So cell-mediated immunity means that we have the T cells, and there's lots of different T cells, and each of them have a different function, and that is how we directly kill and do some things that support that particular actions of the B cells and T cells. So there's lots of subsets, and their actions are dictated by the antigen-presenting cells that activate them. And the T cells require some type of MHC recognition before they can be activated, so that is something different from B cells. B cells do not require the MHC to present the antigen to them. So the cell-mediated, so the T helper cells, we saw these have CD4 markers, and they play this central role in regulating the immune reactions to antigens. And so they activate B cells and other T cells, and some of these involve activating the macrophages. They do that by direct receptor contact and also indirectly using different cytokines. So there's a couple of major types, the T helper 1, T helper 2, T helper 17, and some others. The follicular helper is also a CD4. That's a type of T helper. So the T regulatory cells maintain this happy medium. They control the inflammatory process. They prevent autoimmunity. They make sure that the immune response doesn't inappropriately target the normal microbiota. So these are really important things, and we think one of the ways that, autoimmune diseases may develop is something's wrong with the T regulatory. But we definitely don't want to get rid of the normal microbiota, and we don't want to target it. Otherwise, we wouldn't have normal microbiota. So the fact that they can distinguish between good bacteria and bad bacteria is pretty amazing. So B regulatory cells also regulate the T cells. They become activated and to become plasma cells by cytokines, and the already activated regulatory B cells can secrete their own cytokines that can dampen the T cell response as necessary. Cytotoxic T cells, the third type, are the ones that directly kill. They have this cytotoxicity. They're capable of killing a specific target cell. So the one thing I call these like my assassin cells, because they target a very specific cell type, and that is the cells that are infected with a very specific antigen. And also they can kill cancer cells as well. So, but they have to recognize a foreign peptide complex with an MHC1. So it's not just random killing, it's a really specific targeted thing, and it does require binding to the T cell receptor and the CD8 binding to the MHC that has the antigen in it. And so what they do is they secrete perforins, which punch holes in the membrane, and that causes the cells to leak out, creates a way for the granzymes to enter. Granzymes, just like the name says, sound like an enzyme, and they are. They're an enzyme that attack proteins in the target cells. And so after the perforins and the granzymes, the targeted cells go through cell death by apoptosis. So they basically sacrifice themselves so that the rest of the cells will not be attacked. So the three types of cells that cytotoxic T cells can target are virally infected cells. So they're not targeting the virus, but they're targeting cells that are infected with the virus, and it's very specific, a specific virus. Cancer cells also are targeted by these cytotoxic T cells, and cells from other animals and humans. So they can destroy these cells, and here's an example of a cytotoxic T cells destroying the target cell. So gamma delta T cells. So I wondered, why is it called gamma delta T cells, right? So they have a receptor. They have T cell receptors that actually have, instead of an alpha and a beta polypeptide chain, they have a gamma and delta version, and that's why they're called gamma delta T cells. So they do have T cell receptors. They're rearranged to recognize a wide variety of antigens, so they can specifically and non-specifically. They respond to certain PAMPs or pathogen-associated molecular patterns. They respond very quickly, and they do produce memory cells when they are activated. And so that's like a T cell. It's a specific immunity, but they can also be able to recognize these pathogen-associated molecular patterns, and so that's the non-specific part. So natural killer cells, they are lymphocytes that are related to T cells, and I wondered, why are they called natural killer cells? Well, the only explanation I could come up with was that they kill naturally. Okay, so what does that mean exactly? Well, they don't have T cell receptors, and so therefore they are not specific. So we don't really consider the natural killer cells part of the specific cell-mediated immunity, because they do not possess the antigen receptors that are specific to a particular antigen. But they circulate through, and they can find cancer cells and virally infected cells, and they do kill in a manner similar to the cytotoxic T cells. So they do have perforins and granzymes, so they do kill similarly. But this is a very non-specific killing. So I call these natural killer cells, are kind of like the serial killers, or people that go and not to be insensitive to some events that have been happening this year and every year, but they're like the ones that go and just kill anyone in a mass shooting kind of event. But the natural killer cells are not bad. They actually do help us. And they are the first ones to kill the cancer cells and virally infected cells. They just do it very non-specifically. So as opposed to a assassin cell, they are non-specific in the way that they kill. So what do they have? On their surface, they do not have a T cell receptor, but they do have these NK receptors that are either going to be activating or inhibiting. And so if it's an activating receptor that's bound, then it activates the NK and they will kill. Or they can have inhibiting receptors. If the inhibiting receptors are bound, then they don't kill. And so that is what I have learned about the natural killer cells. But the main idea here is that they kill similar to the T cells, the T cytotoxic, but they don't have a T cell receptor, so they are not specific. And then there's this natural killer T. So this is a hybrid of both the T cells and the natural killer cells. So they do have the T cell receptors and the NK receptors. And so they're stimulated by glycolipids and foreign cells. They produce cytokines, granzymes, and perforins, just like the NK cells and the cytotoxic T cells. And they do trigger self-destruction in target cells. But because they have T cell receptors, they are specific, but they can also be targeting non-specifically as well. So that is the NK cells. So our concept check here for section 15.5 is to describe the function of the T helper 1, T helper 2, T helper 17. And then to second question, cells that regulate T cells are blank cells, and TC cells are blank and blank to destroy virus-infected and cancer cells. And question 4, blank have a similar function to T cytotoxic cells. So question 1, so the function of the T helper 1 is to activate the immune response, and T helper 2 is to activate the eukaryotic pathogens like parasites, the phagocytic worms, or parasitic worms, and protozoans, parasitic protozoans. Also allergies, so those are a lot of functions for the T helper 2. And the T helper 17 is going to be involved in inflammation. So cells that regulate T cell activities are called T regulatory cells, and there are also B regulatory cells. TC cells use perforin and granzymes to destroy virally infected cells and cancer cells. And blank have a similar function to T cytotoxic cells, which would be natural killer, and natural killer T cells also have a very similar function, and they kill in a similar manner. So that's the end of section 15.5.